Okay, so if there aren't any questions, I'm going to go ahead and get into it then. So when we look back at this chart, I want you to remember that the thing I'm trying to emphasize to you with this whole idea of protista and diversity is this kingdom protista is essentially dead. It's kind of sticking around just for matters of convenience. But if you look at this right away, it's very, very polyphyletic. There's all kinds of things that we wouldn't normally call protists that are mixed in with other protists. And not only mixed in, but have ancestry that goes back quite a ways, like red algae, chlorophytes and caryophytes. That's even grayer there. But these critters here are universally regarded as quote unquote protists, or the Archaeoblastida, right? So these are considered protists. But look what's mixed in with there. Land plants are mixed in. And we're saying that land plants and these protozoans had an ancestor somewhere way back here, and that these are also protozoans over here, the sarclade and the excavata, which we talked about the other day. So you can see right away that the kingdom protista is destined for a downfall. It's going to have to be split up. I mean, I don't know exactly how they're going to do it. And if you remember our discussions of evolution, you realize what's going on here. This is those gray areas again, right? This is those evolution isn't set events that occurred. It's gradualism. It's punctuated equilibrium. It's gradual change through time overall. So that organisms are basically, they don't just say one day, boom, here's a protist. It kind of, you become protist-like. And then some of these protists became land plant-like, and some of these protists became fungal-like, and then some of those are fungal animal-like first, and then they split off because the nuclearids down, oh, come on, pencil. The nuclearids down here and the choanoflagellates, those are still considered by many protists-like, or protists, if you will, okay? And look how close. They're close. Choanoflagellates are much more closely related to us than they are to other protozoans. Matter of fact, we're more closely related to fungus, right? I said that the other day, than to other protozoans. So you see what I mean by this being a polyphyletic kingdom. I want you to always keep your mind open about these things, because, of course, taxonomy changes because we learn more and more about these critters. All right, so we know one of the things they do is this. They like to engulf things. We have a lot of evidence. Oh, back one, Steve. We have a lot of evidence of that. I showed you this the other day, this picture in the lower right, a nucleomorph. I don't think I mentioned this. I was looking at this morning. I said, I think I forgot to mention a nucleomorph. The reason I put it in red is what you're looking at is a cell inside of a cell. So the nucleomorph is the nucleus of the cell that was engulfed. And this is an actual cell that's alive today. It's in your book. It's figure 28.4. And when we look at genes from the nucleomorph, it has its own genes. They're still being used. And the nucleomorph genes suggest that that's a green algae inside of there. So this secondary endosymbiosis, like where maybe chloroplast came from, we still have evidence. This is not a fossilized cell. This is not a dead cell. This is actually an engulfed cell that exists today in some green algae. All right, so we have a lot of circumstantial, we have a lot of current observations that support this idea of endosymbiosis. You remember back in the biogenesis, this was how prokaryotic organisms survived. There's a lot of how a prokaryote survived the changes in the atmosphere. Remember, they entered into relationships. Okay, mitochondria, chloroplasts. With the red algae, now red algae we haven't talked about, we'll talk about it today, but they can also photosynthesize. They do it with a little bit of a trick, so it's kind of cool, and it has a lot to do with where they live. But red algae also photosynthesizers, so things that we'll talk about called dinoflagellates, stromatophiles, they also have plastids. They have these red plastids. And a plastid, remember I told you, is a term that refers to these special organelles like chloroplasts. So chloroplasts is the green ones, and then, gosh, I don't remember what they're called in the dinoflagellates, the things that do the photosynthesis, but we'll talk about those in just a second. Oh, so we went through the excavata. Yes, it is. Well, no, not an algae, not an algae. We call it an algae. Remember that word algae, how It is caused by a red thing, by the way. Yes, that's why I immediately said yes. When do we get to the? We're going to be getting to those here in just a couple slides. I'll answer your question. You'll see what I mean by it not being analogy. And I'm glad you brought that up, because you also will remind me to discuss again that idea of what analogy is, so we don't get confused. Where am I? Where am I? Oh, I'm right here. Okay. All right. So we went through the excavata group. Now we're going to move into a clade. Now these names, excavata, the SAR clade, the archoblastida, the uniconta, those aren't necessarily taxonomic names. And until these things are taxonomic names, they're not really official. So we can work with these since we're dealing with this huge polyphyletic group, right? So the SAR clade, the SAR is actually an acronym. The three big groups in the SAR clades, the ones we're going to talk about, I put in the black box you see there, the black rectangle. The stramenopiles, the alveolates, and the rhizarians. Those are the three big groups. And then we'll break them down and look at diatoms. Oh, there they come. The algae, things called the dinoflagellates, the apical complexans. There's a mouthful. Ciliates, which you might say, oh, I'll bet you I know what those are. Those are the things with cilia. The forams, or actually their full name, foram is a nickname. The forams, or the foraminiferans, there we go. Cercosarians, the radiolarians, and then some more algae gets thrown in. And then there's the green algae, which are kind of plants, especially if you're a caryophyte, and these really cool things called slime molds. Oh my God. Yeah. Look on YouTube and look up slime molds or weird thing in the sewers of New York City, and you'll see videos of slime molds. They're the neatest thing. And then other critters called tubulinids, and amoebas, nuclearids, and the choanoflagellates, so mixing in with the plants. Okay, so today we're going to do the SAR clade, or well, actually we're going to start with the SAR clade. Now this is a really large group with lots of organisms, lots of species, but at least we know with the SAR clade that it's all monophyletic. So we're not sure how that all fits with the rest of the protozoans, but we do know at least what we're calling the SAR clade is going to be considered monophyletic. So that's nice. At least we know that we think we can probably fill in, well, some of these, see these dashed lines, that indicates unknown phylogeny there. But at least we can start to fill in and say, when it comes to the SAR clade, we could probably darken in this line and say, at least from there, we might know what's going on with these guys. A lot of them engaged in that secondary endosymbiosis, that slide we just looked at, because remember, you can undergo, well, I'm not going to go all the way back. You can undergo endosymbiosis and then yourself be engulfed. So you can be secondary endosymbiosis. Okay, the name Stramenopile, and this is one of the ways you can help remember what these different groups are. When it comes to the Stramenopile, stramen is straw, it's from Latin, and pila is hair. Remember pili, P-I-L-I, in the bacteria? Those were those adhesive things, the fimbriae they were called, but if they got really big, it became a sext pili. Pili is hair, right? So organisms that fit in this group have flagella that are hairy. They have hairy flagella, right? And many times they'll have two flagellas. One will be hairy, one will be smooth. And down below is really kind of a cool picture. You can see the hairy flagella. It's like flagella on flagella. It increases the surface area. It's so weird. Look at the critter on the left. To me, it looks like a bath sponge thing, right? Looks like one of those bath sponge things that you might put soap on if you're taking a shower. There are some Stramenopile, yeah. There are some Stramenopiles, by the way, that have smooth flagella, but are still considered Stramenopiles. Once again, going back to this idea that, oh man, you know, just because they look a certain way, because we're using this hairy flagella to classify them, right, to call them Stramenopila. That's why it's straw hair. It's hairs that look like straw. But what has happened in those Stramenopiles that have smooth flagella is they have evolutionarily lost it. Their ancestors had hairy flagella. They're no longer around. Now they have smooth flagella, right? Just reinforcing again, even among these protozoans, what it looks like ain't what it looks like sometimes. Okay, so now let's look at a few of these stramentopiles, this group of the sarclade, the diatoms. Oh, these are cool. This isn't artist reconstruction, folks, other than maybe it's been colorized, but these are actual electron microscope pictures of diatoms. The diversity of diatoms there's well over 100,000 species of them, right? 100,000. Now, fish, what do we say, 30 to 40,000 species of fish to kind of give you a comparison. I think mammals were less than 10,000, if I remember correctly. 100,000 species of these guys. All with these different really bizarre shapes. All the well, yeah, there might be some species have the same shape, but a lot of these different shapes. So I've completed or kind of put them in the background too. These are wonderful to look at. They're photosynthetic, unicellular critters, but they fit together like a shoebox. If you can see the two tops, if you can see the two parts of it, and that one on the right, there's this part and this part. If I could draw it, let me get white. That might be easier. There we go. If I could show you what that looks like by the side, the two parts fit together like this. Right, so they fit together like a shoebox, and actually when they reproduce, what they do is they just split the shoebox. I'm sorry, this is not to scale, and then you just replace the other piece. Isn't that cool? Sort of weird asexual reproduction. The shoebox, by the way, is made up of something that's relatively firm, so there's some need to make this shoebox resistant, right, against pressure. It's actually made of silicon dioxide, a major component of sand. But sand, silicon dioxide, glass, it's hard, it's firm, if you will. And if you're wondering, is that predator protection? Yeah, maybe to some degree, but it has a lot to do with where they live, right? And that's what I was saying earlier about the diatoms. It's always so wonderful to me as a biologist to make that, to kind of be aware or make that connection between what a critter looks like, how it works and operates, what structures it has with where it lives. Because I mean, that's why I said at the beginning of the semester, this class is nothing but evolution over and over and over again, right? These organisms, these diatoms, oftentimes many species live in the very deep ocean where pressures can be outrageous. We're feeling about 16 pounds per square inch, and I'll keep it in English systems because we're more familiar with those, of pressure pushing all around us right now, standing at sea level. Here in Florida, we're about sea level. 16 pounds per square inch because the air weighs something. It doesn't weigh much, but there's a big column of air above us, and it's pushing down on us. We call it 16 pounds per square inch. We call it 1 atmosphere. So we're giving it a metric so we can measure it. Now, when you go into the ocean, for every 33 feet, 10 meters, so a little over 30 feet, 30 feet and change, right? For every 30 feet and change that you descend in depth, you increase the pressure by 1 more atmosphere. So you only need to dive down in the ocean 30 feet, and now you're at 32 feet, or 32 pounds per square inch, okay? You were 16 at sea level. You go down 30 feet, you're now at 32. You go down another atmosphere, another 32. Every time you go down 10 meters, 30 whatever feet, you're adding another atmosphere of pressure. Now, if you're a scuba diver, or you've scuba dove, or you've even dived in a pool down to, 15 feet, okay, you felt it, haven't you? You felt the pressure on your ears. Scuba divers, right? That's why you have that thing in your mask, so you can grab your nose and blow the pressure back into your middle ear, because the pressure is pushing on your eardrums. Another reason why, if you scuba diving when you're surfacing, you're taught to never surface any faster than your air bubbles are surfacing, your carbon dioxide bubbles. As you're exhaling, they're bubbling out. As they're surfacing, you're taught to stay at that level, because if you surface too fast, then what happens, the pressure is released too quickly. It can mess up your eardrums, and it could also give you the bends. So pressure in the ocean is no big deal. If you guys know what I'm talking about, right? You felt it in a pool. You felt the pressure on your ears, and the opposite in a plane. The opposite happens. Oops, almost knocked my drink over. Right? Now, look at the pressures that these organisms can live. All right, so we were using what, 16 pounds per square inch. At the depths that they lived, so you can see depth and pressure increases very quickly with depth. They're up to 33 million pounds per square foot. So that's a little bit bigger than a square inch. Oh, I should make the conversion. But 33 million pounds per square foot is a lot, right? That would crush things. Look at those styrofoam cups. Yeah, look at those styrofoam cups. Isn't that neat? It gives you an idea. 33 million. Yes, it is. That's a huge amount. Styrofoam is mostly air. So what they've done is they take them down in these submarines, submersibles, or these other things that go down to depth, like where you would find these kinds of pressures, and they let the ocean pressure squeeze the air out of the styrofoam cup, and they come back like shot glass size. It's really cool. If you ever get a chance, go down to Vero Beach on the East Coast and go to the Harbor Branch Oceanographic Institute. Harbor Branch Oceanographic Institute. If you type up online HBOI, they do oceanographic research, but they actually have a tour, and they'll take you and show you around, and you can see actually some of these cups that they brought back from the depth on there. If you're an aquarist, aquarium person, you're interested in that kind of stuff, you may be familiar with something called diatomaceous earth. And the word diatomaceous is from the fact that that's what diatomaceous earth is made of. It's made up of the shells, these silicon dioxide shells of these diatoms. It's made up of them and then ground up. The reason why it's such a good filter is you can make that stuff really small. It's glass. So you can really small particles do a really good job of filtering. Now the nice thing or the good thing for the ecosystem is every once in a while, if an ecosystem goes through a bloom, which Thomas, right, you asked about a red tide earlier. Yeah, a red tide is due to a bloom. It's like we talked about an algal bloom the other day. It's like a whole bunch of stuff, a whole bunch of algae just reproducing very quickly. Now, red tide is not an algal bloom, but it is a bloom still. Diatoms can actually help with those blooms. See, what happens with those blooms, everybody said, well, wait a minute. If a bunch of algae bloom in a lake and they're and they're photosynthesizing, aren't they making a lot of oxygen? Yeah, that is true, right? If you added a whole bunch of more algae to a lake that were photosynthesizers, they would start really cranking out in photosynthesis and producing a lot of oxygen. But what you're forgetting about the equation, or what people many times forget, is photosynthesizers, they're also, okay? And they're reproducing so quickly that their respiratory rate, sucks up so much of that excess oxygen they're making that the other critters around them, more specific, like the big ones, like the eukaryotes, like the fish, start feeling, oxygen stress. They start having low oxygen levels. Diatoms, what they do is they're really good at being a carbon sink during these blooms. So what happens during these algal blooms is the algae say, wow, conditions are great for reproducing. We've got all these nutrients and carbon is everywhere, because you got to have carbon to, link these molecules together. Wow, we got lots of blah, 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 blah. So what diatoms do is they'll suck up that carbon and go through their bloom, which doesn't cause the problems that we find with, some of the prokaryotes going through a bloom. And so we call them a carbon sink. Carbon sink, or sinks and sources. When we talk about ecosystem dynamics and environmental science, sinks and sources are where something goes, like heat sinks, like the ocean is a heat sink. And the ocean can also be a heat source. So it's where heat may be released from or heat is absorbed by. Okay? So you might discuss the different sink qualities of ice. Is ice a big absorber of heat or a big reflector of heat? So it's not a very good sink. Ice reflects a lot of heat. So it would be a very good heat sink. Okay? So these guys suck up a lot of that excess carbon. So that's good, at least in some degree. Ooh, the golden. Here we are with that word algae. So here's algae number 2, right? We've seen blue-green algae, which were prokaryotes. Anabaena. Anabaena, saw it in class. I don't know why I went Anabaena. Anabaena. I'm sure I was talking about Anabaena. Good morning, Candy. I'm sorry. Who else? Jennifer, I said. Spencer. Yeah. Good morning, Candy. So you all remember the blue-green algae, right? And that was a prokaryote. So here's another algae. This is a golden algae, and this is a protozoan. Okay. Now the color that comes from the golden algae gives it its name. It's a color from carotenoids, and if you remember in Bio 1, carotenoids were an accessory photosynthetic pigment. Okay, not chlorophyll. There's two common ones, xanthophylls and the carotenoids. What color were carotenoids, or what vegetable do you remember has a lot of carotenoids in it? That's how you always remember the color. Who's got carotenoids? Close. Starts with a C, but carrots have a lot of carotenoids. Yeah, they do. That's what makes them orange. If you have a lot of yellow or golden, if you will, then you would end up being orange. Dinobiron, that's a picture of a carotenoid. See, it's got sort of a golden appearance to it. These organisms are biflagellated, and we're going to be moving on talking about the other groups of, protozoans that gave rise to other critters. And this whole idea of how many flagella you have and where they are is going to become important. And if you're wondering why is it so much just about the flagella, it has to do with the idea that flagella are often associated with larval organisms. So similarity, it's kind of like I was talking about heterochrony, developmental genes when we were at the end of the what was it, 26, I think, that chapter, that, things that are similar when they're larvae are oftentimes closely related. So one of the things that we fix on, among other things, is how many flagella and whether they're on the back or the front, okay? Golden algae, the carotenoids are photosynthetic pigments, so they can photosynthesize, although some switch. Mixotrophic is like the euglena, remember? Best of both worlds. You can be photosynthetic. You can also be heterotrophic. And remember, photosynthetic, the key is the carbon source. If you're photosynthetic, you're an autotroph, and the carbon comes from carbon dioxide. If you're mixotrophic and you use heterotroph, then the carbon source is another carbon source. Remember the prokaryotes, the way we break down metabolism is where do you get your carbon to make big carbon molecules, and where do you get the energy to do that? From the light or from oxidation, those other ones that we were looking at. Unicellular, colonial, kind of multicellular, kind of not multicellular. Depends on how you divide it up. Depends on how you look at it. It's not a set place that multicellular starts. Oh, this idea of cysts, which we've seen more than once. We saw it with the anthrax spore, didn't we? Remember anthrax spores? Why do anthrax spores make cysts? It's kind of like, why'd the chicken cross the street? Protect themselves, true, against what? It kind of hints there, too. What are you protecting yourself from? Spencer? Nicole? Start calling you guys. Sydney? Yeah, attackers, but since cysts tend to be very persistent, and since they tend to be formed in such a way so they are resistant, then we generally associate organisms that form cysts with a way of waiting till the environment becomes better. So not so much predation or prey protection, but more along the lines of, wow, if the environment starts to deteriorate, I'll make this relatively resistant structure, this cyst, I'll form it, put it out in the environment. Probably I'll put a lot of them because most of them probably won't live. And then maybe sometime in the future, it's kind of cheating a little bit with natural selection. It's kind of avoiding adaptation. It's kind of like the old ostrich putting its head in the sand. Oh no, the environment's changing. I'm going to make this thing that hopefully can survive, and maybe the environment one day will become good again, right? So it's kind of like the environmental or the evolutionary equivalent of doing that, right? So these organisms, if the environment deteriorates, they can form those cysts as well. And again, the key with cysts is everything is numbers too. If you make a lot of them, then a lot of them are going to survive. Now, before this class, I bet I was going to say my paycheck, but that's not a whole lot of money. And I need it myself anyway. But I'll bet that if you asked 100 people what a seaweed is, they would tell you it's a plant, right? How many of y'all honestly would have, before I said something, would have thought that a seaweed, what we call seaweeds, the big things of kelp, is a plant? Yeah, I would have. Certainly looks like a plant. It looks like a plant, doesn't it? It's got all the same. Yeah. So if you took a marine science course, but I mean, other than that, it looks like a plant. So yeah, let's go to the next slide. Then I'll come back to this. So if we take a look at a seaweed, this is a brown algae. I think this is fucus. I believe this is fucus. I'm not sure. We'll talk about those in lab. But if you notice, they have all the parts of a plant, don't they? They have something that holds on. It's not called roots, though. Now, oftentimes it'll be referred to as roots, but you guys know better than that. You know that true roots, or you're going to learn, of course, that true roots, so you would know better than that. But true roots, or, well, you know technicalities, are only found on plants. True roots. That the thing that an algae makes, since it's not a plant, although some of them are kind of related to them, anyway, the roots of a brown algae are called roots. They're called a holdfast. It does the same thing. It holds them tight, but it's not called roots. Okay, you might see it called roots, and it's not wrong, really. It's more like being more right. You see what I mean? The stem of a plant would be the stipe, and the leaves would be blades. Okay, so answer the question for me in the upper left. The roots, the stem, and the leaves, if you will, all in quotes, of this brown algae, of this protozoan, is that a homologous trait or an analogous trait? See what you remember from the first group of information. Homology. I'll do the Jeopardy thing. Homologous or analogous? Don't split hairs with me either. You know that, we can argue all of it at one point, but you know what I'm talking about. Okay, anybody else? Gabriella came in. Let me put that in there. Anybody else? Come on, guys. Just type in the first letter, H or A. Candy, what do you think? Jennifer? Okay, Candy says analogous. Jennifer says, everybody's got a question mark. Okay, all right. Spencer, Sydney, Nicole, how about y'all? Oh, Gabrielle left. I didn't want to get called on. Nicole says it without a question mark. Is that because you're sure, Nicole, that it's analogous? Yeah, I got you. Okay. Can you explain why you came up with analogous versus homologous? No, I bet they don't share a recent common ancestor. As a matter of fact, yeah, they aren't, Spencer. They aren't closely related to one another. Remember, I said don't split hairs because everything's essentially homologous. But what we mean by homologous is the similarity in structures due to common recent ancestor. If it's analogous, it's going to be common function. A holdfast holds protozoans, which aren't all that related to land plants at all, down. A stipe allows the other part to stand up so that the blades can, guess what? Photosynthesize. Okay, so these things can photosynthesize. So they just use the same kind of structures because they're dealing with the same kind of environment. There are even underwater plants that grow this way that are true plants with roots and stuff like that. Okay, yeah, good. All right, so let's go back to that first slide. They're kelps are not they're not plants. They're seaweeds, and we often use the word weed for a plant. But they're not, right? They're not plants. Many times they don't grow in really deep environments. These kelp forests that you see here on the right, I used to dive those when I was in California. Water was just gosh cold. And sharks love to run through the kelp forest attacking prey. So it was always really interesting. It never happened to me, although I wish I had but buddies of mine would be diving in these kelp forests and they would be so thick. And the sharks, what they do is they try to do ambush predation. You know, they can't really see where they're going. They're just hoping they're going to jump into the kelp beds and something will be, whoa, it's surprised and they can grab it real quick. So they'll zip, the mako sharks, which are like mini great whites, will zip through the kelp beds and hoping to, you know, shock something that's, you know, some fish that's kind of out of position, especially the young fish. It's the same thing that this beluga is doing down here. All these are this is all kelp beds, and what it's doing is waiting for a fish. It's too big to just go darting through, so it waits for someone to kind of get a little too far out, you know, like, wow, and grabs one. But buddies of mine that diving say before you be in a kelp bed where you just barely can see anything, like in a dense forest, and phew, you know, within just a few feet away, a mako shark comes whipping through. Probably doesn't see you necessarily, because it's, you know, that would be a cool experience though. So, yeah. My son's going to Australia here in February. He says he's going to go shark diving. He's going to try to go white shark diving. Technically, great white sharks are extinct. That's the ones with the big, huge teeth that you can buy sometimes. The ones around today are technically just called white sharks. How cold can kill? I don't know. Pretty cold, because the water in California was, I don't think it ever got above 68, 65 something degrees, and it gets a lot colder than that in the winter. And where California is, being on the west coast, the water's coming down off Alaska. That's how the water circulates. Here we're lucky, it comes up from the equator. That's why we're nice and warm. But there it's the other way. The beaches in California, when I was out there, oh man, they're freezing. You hardly ever win the water. So back to my little statement up there. Why do you think these things grow in relatively shallow water? Yeah, they got to have sunlight. Sure, absolutely. Now, yeah, absolutely. Now, they're all multicellular, but again, I want you to treat that word always with the idea that there's more than one definition for it. You may even read it in a science book about multicellular brown algae. But what I'm trying to say is it's not a right or wrong. It's just a matter that folks differ on where that truly happens. Okay, now I want to talk about a form of reproduction that maybe you guys saw. You might have saw it in Bio 1, or maybe you were introduced to it, but you're going to see a lot of it. You're going to see a lot of it in Bio 2. The organisms that I'm going to talk about that do this form of reproduction are the algae that I was just showing you in this picture here. So that is Laminaria. But they're not the only ones that do it. Plants also engage in this type of reproduction. Other protozoans do. Fungi, there are many fungi that engage well, no, wait, that's not exactly technically true. But anyway, there are other plants that engage in this type of reproduction. There are other fungus, or I'm sorry, other protozoans that do, not other fungus. That's not, is it? It's kind of technically, anyway, I don't want to confuse you. So let me talk about this type of reproduction. The name of this type of reproduction is called alternation of generation. And where the word comes from, let's talk about that first, because that's going to help us understand, is what we're talking about alternation of generation is in alternation of generations, organisms that use this type of reproduction have two, not one, but they have two multicellular generations. And when I'm talking about generations, I simply mean haploid diploid. That's all. So an organism that has alternation of generations has a multicellular diploid stage. Okay, there it is. There's diploid. And we call that a sporophyte. And they also have a multicellular gamete stage or haploid stage, which we call gametophytes. Okay, now, how is that different in the way we reproduce, us animals? How is that different? You might be hearing my dog snore in the background if you're wondering what that noise is. Okay, we're haploid. They are too. Gametophytes are N. They're haploid also. But what's different about... It's also different. Remember from your Bio 1, folks, in meiosis, we have haploid and diploid stages, right? Sperm and egg are haploid. Somatic cells, liver, kidney cells are diploid. So, we turn diploid cells into haploid cells in meiosis, and then we turn haploid cells into diploid cells when fertilization. And they do too. They have meiosis and fertilization. You can see it on the screen here, but again, what's the big difference between alternation of generation reproduction and the way we do it? Anybody can help Thomas out. Are sperm unicellular, or are they a group of sperm? Are eggs, when they're ovulated, unicellular or groups of eggs? Sperm are unicellular, aren't they? Right? They don't exist as a mass of sperm cells. They exist as unicellular cells. So we make unicellular haploid. Okay? These critters that reproduce through alternation of generations, they make multicellular haploid. They make gametophytes. You see, look at how the reproduction occurs. First of all, we'll start with meiosis, okay? So, and pay attention to the colors. We're in the blue. So we're in the haploid part, right? That's what meiosis does. Meiosis divides chromosomes in half. Don't forget that, okay? Mitosis makes more, but it doesn't divide the number of chromosomes. It also, makes variability. You understand all that. But anyway, in meiosis, we produce spores. Spores are haploid. They're called zoospores, not because they have anything to do with animals, but because they're flagellated. And we generally associate movement with animals. So things that move, that have flagella, in the case of spores, we call them zoospores. There are other kinds of spores that don't have flagella, that are kind of moved passively on the wind. But zoospores, obviously water's involved here, can swim, okay? And what happens with zoospores, though, is they don't, from the haploid, immediately go out and find another zoospore and engage in fertilization. No, there's a big thing in between, notice. Zoospores germinate into haploid haploid multicellular gametophytes. See, zoospores aren't gametes. Zoospores are, in some cases, more resistant structures. And what they do is, when they find the right kind of environment, is they don't fertilize, they germinate. And when they germinate, they make these multicellular gametophytes. Now, we don't do that, right? Our zoospores, if you will, are germinal cells, are gametes, egg and sperm. And when they come together, it makes junior, it makes another offspring, okay? They don't do that. They have the step in between. Now, if you're thinking, okay, well, why? And you should always be thinking why when you go, wow, that's a wholly different way than we do it, okay? If you think about why, think about what happens to most organisms' gametes, whether they reproduce this way or the way we do. What happens to most organisms' gametes in the environment? Yeah, they don't make it, do they? They don't make it. Right? If we were talking about sea urchins, for example, sea urchins, when they reproduce, what they do is the male and the female will actually just, the male will spawn and deposit sperm into the ocean, and the female, through chemical signals, by the way, that's why I do it at the same time, she spawns eggs, a bunch of sperm, a bunch of egg, and they float in the ocean together with the fish just going, oh my goodness, it's a golden corral bar. Man, they come moving in, attacking all the eggs, and all the eggs aren't very big, so even the zooplankton get in there, and many of them wash away and never come into contact with one another, or they finally find each other, boom, along comes a fish. So how many eggs do you think sea urchin and how many sperm do you think sea urchin put out there? Someone else help Thomas out in participating here, okay? Candy, Spencer, Sydney, come on. Let's all join in here. What happens to most of sea urchin spawn and eggs, do you think? Eat and die, don't make it, float away. They certainly don't become junior, right? So how can you better your odds of getting junior if most of those egg and sperm are not going to come together, are going to be, destroyed somehow? How can you increase your odds? How do bacteria do it? Make lots and lots and lots. You got it. Yes, you make a lot. So a gametophyte is a multicellular gamete producing factory, you see. And each of those cells can then undergo mitosis, right? Each of those cells in that multicellular gametophyte, hundreds, thousands, whatever cells big it is, can undergo mitosis and make boatloads of gametes, you see? Tons of sperm, tons of egg, with the hope that at least some of them do the fertilization thing. And you're back into the zygote, just where we are, right? And now we jump back into where we start. Isn't that cool? And then as a zygote, you undergo mitosis again, and you get bigger. Right? That's what's happening there. But notice your diploid. Isn't that cool? I love that. That's so neat just to think about what a clever way. And if you're talking about organisms, and I'm kind of jumping ahead of myself in the evolutionary idea, but if you talk about organisms that may have gave rise to critters that could make it out on land, even just in water, okay, but make it out into that harsh environment, if those critters that might have excelled, at least some of them were ones that made lots of offspring, okay? All right, so a little terminology before we move on. Gametophytes are called gametophytes because they make gametes, right? Egg and sperm. Sporophytes are called sporophytes because they make spores. The root phyte, P-H-Y-T-E, is roughly associated with plants. And although in this case we're not talking about plants, there are epiphytes. Orchids are epiphytes. They grow on other plants. Maybe think of it, a phyte, as being a thing, but more like a plant thing. Forget it. Sporophytes make spores, gametophytes make gametes. Okay, now, in this particular case, this organism, Laminaria right there, has what we call heteromorphic generations. Look on the left, you see what I'm saying, and you look at it right away and it makes sense. If the generations are not similar, if they're different, they're heteromorphic. If what the sporophyte and the gametophyte look like are similar, then it's isomorphic. And we'll see isomorphic ones, I think, when we get into the plants. Okay, so that's alternation of generations. Anybody got any questions on that? That's an extremely important part because that is going to fit in very nicely with this idea of evolutionary change in organisms. That if you generate a lot of offspring, if you do a lot of meiosis, you generate a lot of variability, generate a lot of offspring, you hedge your bets against that really nasty old world out there. Okay. Whoops. Good. All right. Let's go back and orient ourselves where we are. I think we're in the alveolates, aren't we? Okay, so now we're going to be jumping out of the stramenopiles, moving to the next major group in the sarclade, the alveolates. Now, alveolates are all unified by one thing, sort of like the stramenopiles had hairy flagella. The alveolates all have, there you go, they all have alveoli. Okay, no kidding. That's why they're in the alveolates. Now, an alveoli is a general term. It doesn't necessarily mean to just protozoans. Us animals, we have alveoli, right? Where would I find our alveoli or an alveolus? Where would I look? Yep. So what is an alveolus in the lung, Spencer? Yeah, it's an air sac, okay? Look at the alveoli in the picture. What are they? They're sacs, aren't they? Flattened vesicles, okay? If you want to be completely technical about it, flattened vesicles, but they are sacs. Now, we don't know exactly what they do. We don't know exactly what those sacs do. So it's another one of those little mysteries about protozoans. We know that all members of this group have them. The group I'm going to talk about members of the group here in just a second. We know that they all have these sacs, but we're not sure what they do. Now, there is one group where we suspect what they might do, but I'll wait to talk about that when I get to it. They might work like Golgi bodies, okay? And you remember what Golgi bodies do is they export material. All right, so think about exporting material out of the alveoli, okay? You can see the flagella. Now, this is now a different kind of flagella than we saw in the prokaryotes. Remember, the prokaryotes had that spinning, really cool kind of flagella. This is a different kind of flagella. This is the eukaryotic flagella. Oh, and there's the dinoflagellates. This is the red tide, Thomas, that you were talking about. This is what causes the red tide. Dinoflagellates. So it's not an algae, okay? It's not an algae per se. It's a dinoflagellate. But it is red, though. It's red in color. When it blooms, it forms the red tide. And the problem with that is it releases toxins. These organisms release toxin anyway, but in blooms, they're releasing a lot of toxin in a concentrated area. That's why organisms can die. If you've ever been at the beach during a red tide bloom, you might have even felt it. It affects the respiratory system. So you might have felt it in your lungs, like making you cough and stuff. The dino part comes from the fact that they have these two flagella and they whirl around. They're whirlers. So dynamo. So they can whirl around, mostly photosynthetic, although not all of them use chloroplast to photosynthesize. They're unicellular shells again. So they have shells like the diatoms did, but not made of the same material. This time made of cellulose. That's kind of cool. Who else has shells made of cellulose? Yeah, it sure can, especially if they eat fish that have incorporated the toxin, but can also affect them in their respiratory system as well. Absolutely. Right, so cellulose, folks. Who else has cellulose? Right, they do. And so does this indicate homologous or analogous, would you say? Absolutely, right? Analogous. The fact that cellulose is just simply analogous in this case. Glucose is not unique, which is what cellulose is made from, is not unique just to one group of organisms. You're going to find it in other organisms too. Most of the time, dinoflagellate, well, not most of the time, all the time, they're considered planktonic, and the word planktonic refers to a lot of different types of organisms or a lot of different kinds of plankton out there, but technically planktonic just means that you float. Planktonic organisms are floaters. I think it actually comes from the Latin for drifting. The other type of organisms in the sea that can swim, the fish, even the marine mammals, are called nectonic, or not nectonics, nectonic. There we go. Dinoflagellates are a lot of those ones that can luminesce, right? Sometimes they have symbiotic bacteria down in their tissues, and when you agitate them, the bacteria fire off, and they emit this fluorescent glow. Oh yeah. Beautiful at night. I know when I was in the Navy, sometimes especially if the ocean was cleared at night, the ship going through the water, as it pushed out the wake, oh, it all glows. Incredible. Different colors to greens and blues and all kinds of neat stuff. How much did I give myself? Did I give myself just for today to doing the? We're gonna have to do more protozoans next. That's okay. We got plenty of time. All right, next group. So we're still in the alveolates. Now this is the one where maybe those alveolate, those sacs, might have something to do with what the cell works in. Apical complexins are animal parasites, and the reason why we call them apical complexins is because the apical, and an apical is basically just a point for the most part. For example, in plants have an apical meristem. So they have an area in the root that's right at the point, okay? So apical is point, or another word is an apex. In the picture, they're calling it an apex, okay? At the apex, or at the point, there is this complex of organelles, an apical complex. And involved in that apical complex are some of these alveolates, some of these sacs. Remember I said that they act like Golgi apparatus. So what they might be doing is secreting material that allows these, in this case, in the picture, these merozoites, which are in the life cycle of a parasite. It's allowing this merozoite to drill its way through the membrane of the host cell, which in this case is a red blood cell. So in the case of the apical complexins, being included in the alveoli group because of the alveoli, we think we may have an idea what they're doing with those alveoli. They're using it to get into the red blood cells. The merozoite that you see in the picture here comes from the apical complexin that causes the malaria disease. The organism is known as Plasmodium. It's a protozoan. Now later on we're going to be talking about plasmodial slime molds. It's not the same thing. Plasmodial slime molds has to do the word plasmodium with the way they move, okay? So don't get those confused. Here we're talking about a protozoan named Plasmodium that causes malaria. The vector for malaria are mosquitoes, okay? But the organism that causes the disease is known as Plasmodium. I'm going to show you, it's really kind of neat too. It's got a very complex life cycle, and a lot of parasites do. It's very interesting. I studied a parasite in South Carolina that used fish, ducks, and snails as intermediaries. In other words, what happens in these parasites, and we'll see it again when we talk about flukes in animal group, each organism, each host is needed for one larval stage to switch into another, or for one infectious stage to switch into another, okay? So they'll have sexual parts to their life cycle, asexual parts, and they also, as I mentioned, they'll have different hosts that are necessary, they're required. Oftentimes, parasites will die off in an area who use this kind of reproduction simply because one of them dies off. Like in my case, the snails would die off, right? So all the parasites would die off. It almost seems counter to evolution, right? It seems too specialized. But remember, you can be specialized. You do run the risk of going extinct if it changes because you need really, sensitive environment. But you can become very good at it. Okay, so you kind of balance it out. Now, we've gotten rid of most malaria. It's not quite the scourge it used to be, getting rid of the mosquito by using DDT until we found out DDT was a problem because it made bird shells too thin. The fish that the birds would eat, the fish would have DDT in them. It would get magnified in the bird, and that would cause the bird's shell gland, which lays the shell or forms the shell, to make it too thin. So when the birds would lay their eggs as they're moving around and incubating, they would oftentimes break them too early, and then the embryo wouldn't develop. So we outlawed DDT. Still used in other parts of the world, though, and now we've found strains that are DDT resistant. So that's a problem. But drug development and vaccines have helped eliminate most of the malaria problems. A lot of times malaria nets or mosquito nets are given to indigenous folks, say in areas that have really bad mosquito problems, oftentimes in Africa. And one of the problems they found out is not understanding what's going on in the area completely. It's kind of like a buddy of mine joined the Peace Corps and went to Ecuador to help local farmers increase their catch. And when he came back, I asked him, I said, how did they accept you down there? And he goes, kind of like maybe we would accept somebody from another part of the world coming to the United States and telling us how to do something we've been doing for 200 years. He said, at the time. So when they delivered these mosquito nets, they found out that the folks weren't actually using them as much as they were selling them so they could use the money to buy food. Because I might get malaria from a mosquito, but I am going to starve to death if I don't eat. So that's kind of not understanding, what these people are going through. All right, so let's look at the life cycle. So in the upper left is our buddy, the Anopheles mosquito. Right? No, there's another S in there. Oh, forget it. Another S in there somewhere. Okay, so there's the P-H-E-L-E-S. P-H-E-L, not an O. That's how you spell it. All right, so it gathers a blood meal, from somebody that's infested with this, and these things develop. So I'm starting up here at the. Where am I starting? Where should I start? I'm going to start inside the mosquito. Okay, so inside the mosquito, there are these things called sporozoites, and sporozoites are a larval form, more like infectious stage of the Plasmodium parasite, the parasite that's going to cause malaria. Okay, they're sporozoites, and sporozoites develop by meiosis, so they're all genetically different from one another in something called an oocyst, and the oocyst is actually found in the gut wall of the mosquito. So in the gut wall of the mosquito, there's an oocyst. It undergoes meiosis. It develops sporozoites. Mosquito lands on you, injects you with its proboscis, and then injects a little bit of that anticoagulant material, it's saliva, more or less, into your blood so it can gather a blood meal. And in that saliva are sporozoites. All right. Now, sporozoites go around your bloodstream until they find liver, because they like liver cells, and they infect liver cells. So that's why maybe we call them an infectious stage, right? Instead of maybe a larval stage. Notice all of this is still happening while they're haploid, right? Sporozoites are haploid. Notice the colors, right? We're in the blue part of the cycle. Now, merozoites, what they do is inside liver cells, sporozoites transform into merozoites. And merozoites divide and divide and divide, and we get tons of merozoites, and eventually merozoites burst out of liver cells and infect red blood cells. Now, look at that life cycle. Not only do we have different, we have a mosquito host and a human host, we have two parts in the human that host it. Now, in red blood cells, merozoites reproduce asexually, making tons of those, and those keep infesting other red blood cells. And about every 2 to 3 days, a person with malaria feels these fevers and chills. You remember Wallace, right? That's where he said he came up with the idea of natural selection, was by going through these malaria fever chills episodes. And the lysing of the red blood cells, that's what causes the fevers and the chills. That's the symptom of that. Now, not all of those merozoites, or not all of those cells that come out of the red blood cells are merozoites, because we got to complete the cycle here. Some of them are gametocytes. Now, mosquito lands on the human again. Wouldn't it be nice if we only got bit once? Everybody knows you're going to get bit more than once. The mosquito that lands on there, now as it's withdrawing the blood meal, absorbs some of the gametocytes. Gametocytes develop into gametes, and look at the male gamete there. The male gamete don't mess around. The male gamete is not a star-shaped thing. Each one of those little worm things is going to become a gamete. So we're going to get multiplication and become multiplication, blah, 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 numbers again. And then we're back to fertilization, right? The only diploid part of this stage is that short-lived zygote. That zygote that develops inside of the mosquito's gut wall, right? That zygote that develops and then eventually creates an oocyst. So zygote develops an oocyst. Now, look at what they're doing here. What are they using diploid for? What are they using that small diploid stage for? They're using, the malaria parasite. It's using meiosis for variability and numbers, because during that time, it's also going to produce a lot of them, right? And what did these sporozoites actually move to the salivary glands of the mosquito. That's how they get into you, okay? But what is it using the diploid stage for? It's very short-lived, but why does it even bother? Right. Okay, to force differentiation, right, Spencer? Right, to shuffle up genetic information. There are several ways you shuffle up genetic information. One is during meiosis with crossing over and the way the chromosomes line up in metaphase 1 and in metaphase 2. We talked about all that. Another way you do it is by fertilization, because it's random which gametes come together. So that short little diploid stage of the zygote there is just there to increase genetic diversity. Because then you turn around into meiosis and you create more genetically diversity in each of those sporozoites, okay? The merozoites down there between in the red blood cells, they're reproducing asexually. So they're going to be clones of one another. Their job is just to make more merozoites and infest more red blood cells, and eventually some of those turn into gametocytes, okay? Isn't that neat? What an interesting life cycle. What interesting life cycle to ponder how the heck this thing evolved, right? Yeah, we're not going to get through protozoans today. I'm sorry I get a little carried away on the protozoans, but we'll finish it. Don't worry about that. Okay, let's talk about the last group in the sarclade, oh no, we're not, yeah, we are. No, we're still in the alveolates. Another group of the alveolates are the ciliates. Got lost. And the ciliates are called the ciliates. Yeah, I would like you to know it, yes. The ciliates are called the ciliates because they have cilia. And cilia are just flagella that are shorter, and usually organisms have many cilia. They'll generally only have 1, 2, or a handful of flagella, but ciliates have lots of cilia. This is your typical paramecium. It always looks to me like it's got the shape of the bottom of a shoe, right? There's the cilia. They have a contractile vacuole, which we talked about, right? Contractile vacuoles are necessary because they live in what type of environment? Organisms with contractile vacuoles live in what type of environment, tonicity-wise? Contractile vacuole would get rid of excess water because when it contracts, it squeezes the water out. So not hypertonic, right, Spencer? Yeah, hypotonic, right. So, yeah, it's all right. By looking at an organism, you can make assumptions about where it survives. It's got a mouth, of course it eats. It's got the oral groove. It's really cool. Sometimes in videos, you can see them turning. You can see the oral groove. You got food vacuoles. Where I want you to focus, because we're going to talk about variability again here, folks, is you notice it's got 2 nuclei. It's got a macronucleus and it's got a micronucleus. Now, the macronucleus, the bigger one, obviously, the macronucleus is where most of the day-to-day activities, the essential life functions go on. The micronucleus is involved in conjugation. Now, we talked about conjugation in a virus. I'm sorry, we talked about conjugation in prokaryotes, how prokaryotes can form that sex pili between them. Remember, I called that conjugation. So this is the same sort of idea. We're going to be swapping genetic information. So this is kind of sexual reproduction, but not exactly. Remember, sexual reproduction is the mixing of genetic information. That's really what it is versus asexual, but it's not exactly the way, other sexual reproducers engage in it. Let's start with the number 1 at the top. We don't have sexes in paramecium. That's one of the reasons why we don't really call this sexual reproduction, because we have things called mating types. There are no genitalia in paramecium, okay? So we have 2 mating types, and we have the dark purple mating type and the red mating type there. You can see a picture of them together. This is 2 paramecium in conjugation, okay? So we're going to start right past, no, let's start right what's going on right here. Okay, so 2 paramecium of different mating types come into contact. The micronucleus in each divides by mitosis, or by meiosis, which generates variability. And if you wonder where they get 4 from, remember in meiosis, 1 cell divides into 2. We don't have to worry about polar bodies here. 2 cells divide into 4. Okay, primary and secondary spermatocytes, even in primary and secondary oocytes, except for the polar body. When you go through meiosis, 1 cell makes 4. Okay, so what we ended up with through this process of meiosis is genetic variability. Now, of those 4, 1 randomly degenerates. More variability. Okay, more variability as to which one randomly degenerates. The remaining one then divides. Why? Why did you go through meiosis, get rid of the 3, and then divide again? So that at the bottom we have 2. Well, you went through meiosis to get variability. The one that survives the degeneration, you want that to be variable. And then the reason why you divide them into two is because you're going to conjugate. You're going to swap with that other organism. See, right past the conjugation, the part in red, right after they undergo mitosis, the one that had the two red micronuclei swaps with the purple. Now they each have a different colored micronuclei. The two micronuclei then fuse, okay? So now the micronuclei, which were haploid, are now going to be formed back to forming a diploid micronucleus. The macronucleus that was originally around disappears, and now the micronucleus divides into a macro and a micronucleus. Isn't that neat? And what you're doing here is shuffling up genetic information. You're shuffling it up during meiosis. You're shuffling up when they degenerate. You're shuffling up as to which two paramecium come into contact. And then you are shuffling up these two organisms' genetic genome between each other during conjugation. Brilliant. That is just brilliant. And that's what it's doing. Its all purpose is just to shuffle up genetic information because you need variability, right? I mean, I had to have said it at least 100 times by now, that variability is important for organisms. Without variability, you get nothing else. Okay, let me see if I can get through the rhizarians. Yeah, the next group. So now let's move to the last part of the SAR clade. Now the R in the SAR clade are the rhizarians. And the rhizarians include many species that are amoebas, but the word amoeba is a little tricky. Just because you're an amoeba doesn't mean that you belong in this group. The word amoeba is not a monophyletic term, is what I mean by that. Rhizarians all do share something similar. That's, of course, understandable from taxonomy. What they share, among many things, the big thing is pseudopodia that are thread-like, like you see in that picture there. So you might remember pseudopods from amoebas. They were more blunt-like. And in the rhizarians here, we have thread-like pseudopods. A member of the rhizarian group are the radiolarians. Now look in the upper right. Notice there they are. Now what's going on in the radiolarians is they also have a skeleton. They also have a protective covering over them, right? But in this case, it's made of silica, just like the diatoms did, right? The glass. So this is the silica protection for this particular radiolarian. This is all the silica protection. And these things sticking out are these thread-like pseudopods that these organisms have. Okay? Radiolarians probably got their name because they might look like a radio spike, I guess. But what the pseudopods are used for, it's really kind of cool. These are marine organisms. They live in the ocean. These pseudopods are used, number one, for capturing prey. Okay? Organisms swim into the pseudopods, and the pseudopods surround, and then they can digest them, right? So that's pretty effective. But the other thing they do is, and you probably have sensed this yourself, floating water, like especially out on the ocean, you can float on your back the more surface area you present, right? So you kind of put your arms out, and you put your back out straight, and you lay as flat as you can because you increase drag. So that's why you don't sink. So these little thread-like pseudopods that you stick out, not only do these organisms, not you stick out, but radiolarians stick out, you're not a pseudopod. But the reason why they do that, or one of the reasons, in addition to catching food, is it helps them stay floating. Isn't that neat? They don't sink. The little pseudopods add drag. How many of you have ever seen a foram? That's the next group. Or a foraminiferan. How many of you have ever seen a foraminiferan? Anybody? What do you mean maybe? Spencer says no. Everybody else, let's have a poll. I want to know how common foraminiferans are. Candy, Jennifer, Marcelo, Nicole. Jennifer's never seen one. Sydney, never seen one. Nicole, have you ever seen a foraminiferan? No. What if I told you you had? What if I told you, well, oh gosh, I just put my foot in my mouth. Maybe you hadn't. I keep thinking you guys, when you went to school, they may not have had them. All right. Foraminiferans are what chalk comes from. All right. The old white chalk that they use on. When I first started teaching this class, I used chalk. Can you believe it? I actually use a stick and a piece of on the back of a shovel. No, yeah. Foraminiferans have thread-like pseudopods. There's a picture of one down below, right? In these cases, though, they have a different shell. Now, a shell can also be called a test. I know I'm running up against the time, so be patient with me, folks. A shell and a test can refer to the same thing. It's a synonym. But in this case, their shell is made of CaCO3, calcium carbonate. All right, and calcium carbonate is what chalk is made of. Again, they use their podia, their thin podia, for swimming and for feeding and for floating. They also have a life cycle with haploid and diploid generations. The White Cliffs of Dover, that's what you're looking at in the picture there. That's what those are. The White Cliffs of Dover represent the bottom of the ocean, and all these forams are marine. By the way, they don't photosynthesize. They're heterotrophic. Sunk to the bottom after years and years and years, their bodies, if you will, form this cliff of chalk, and then through geological processes, this thing was lifted up. So that's that's what chalk is. Chalk is a foraminiferan. Will you let me finish one more? I know I'm at time, but let me finish the last one in the group so we can start at a good place. The cercozoans. Really neat group, very diverse group. They're found everywhere, the soil, fresh water. They can move around through flagella or pseudopods. What's interesting about cercozoans, and the reason why I wanted to finish up with these, is they tie back to this idea of endosymbiosis. This is Paulinella chromatophora. Okay, isn't that a cool word? Remember I said oftentimes if you just sound the word out, Paulinella chromatophora. A chromatophore is that green thing inside. So these things photosynthesize. That chromatophore is surrounded by a peptidoglycan membrane. Now, remember, prokaryotic cells, whether gram-positive or gram-negative, both have a peptidoglycan membrane. And eukaryotic cells do not have peptidoglycan. It's only found in prokaryotic cells. It's only found in bacteria, domain bacteria, not even in archaea. Right there indicates that chromatophore, that functioning photosynthesis thing in Paulinella chromatophora, was probably an endosymbiotically obtained plastid, something that could photosynthesize. There it is, right in your face. Okay, all right, so any questions on that? We're not done with protozoans yet. We'll have to squeeze the rest of them in, but we will. Don't worry about that. So y'all have a good weekend. Let me stop the recording.